So our next speaker is Nuwan Nanayakkara, and so he's a scientific associate at Rotman Research Institute at Bakerest Health Sciences.
Thank you, Olivia. I think you can see my slides.
Yeah, we can see your slides.
Okay, thank you. So I will talk about our robust method for estimating CVR dynamics from breath-hold wall data without end-tidal carbon dioxide recordings or any other equation. So in summary, we all heard that the CVR provides a sensitive disease biomarker sometimes and can be extracted from bolt response to a vasoactive stimulus such as a breath-hold task. But existing method estimate CVR from bolt data using a general linear model based regression or a correlation with the vasoactive stimulants measured from either end-tidal CO2 or from a study design. And also there are some studies reported modeling CVR from sinusoid or a Fourier series as well. But these are questionable, especially when the subject cooperation is uncertain or when the performance, when the end-tidal CO2 recordings are not available. On our robust estimation for CVR, what we propose is to use frequency spectrum of the whole data without modeling or correlation with other signals. So we are using a data-driven CVR frequency estimation. So avoid biases due to subject performance or regional delay variability. The estimating CVR time delay differences will need a reference signal. So we are using a phase-based approach with a reference signal. In this case, we use a respiratory belt signal, but it can be any non-neural biological signal. To detect the breath-hold frequency, we use the information from breath-hold paradigm. That is, the one we use is 50-second breath-hold followed by 30-second resting and 2-second exhale. It's total of 47 seconds. So this was repeated 6 times. And we could estimate the bolt response using sinusoidal at a frequency, breath-hold frequency of 1/47 seconds, which close to 0.0213 Hertz. Now, this breath-hold frequency is not used as it is, but it is used to search the corresponding bolt responses to find out the actual breath-hold frequency that the patient followed within a certain range that is allowed within a band-pass filter, allowing about 30% plus or minus this time period. Breath-hold frequency detection is done using the frequency spectrum. As I said, if I look at a single voxel, spectrum is look like now the blue one is the pre-processed bolt, and green line is the band-pass filtered bolt signal. As we can see, our band-pass filtering actually reduced the signals away from the window that we are searching for, but did not affect the window within the search area. And what we do is we find the maximum amplitude and the corresponding frequency. Then we do this for all cortical voxels and find the histogram so that we get the maximum occurring frequency corresponding to these maximum amplitudes of the spectrum. And we select this as the breath-hold frequency, assuming that is the frequency that the breath-hold action is performed by this particular patient. This was extracted from the bold response itself. Then after we selecting this frequency, we take the amplitude of the frequency spectrum at each option at this frequency as the CVR amplitude, which is given as the percentage volt, and the CVR time delay is calculated from the from the phase difference with the reference signal at the same frequency and converting it to a time delay by dividing by the angular frequency. So to find the then when we look at the resultant CVR response, we could see it's nicely following the filtered board signal, and the dotted line shows the respiratory signal that we extracted from respiratory belt data. This is an example reference, but it can be anything that is any non-neural signal that can be extracted. But this respiratory signal, our recording is something like blue, but we detect the envelope. Envelope is corresponding to the respiratory pattern that the patient followed, which was shown in the previous graph. Now this detected envelope is spectrum. The phase angle of this detected envelope spectrum at the base breath-hold frequency is selected as the reference base to calculate the CVR time delay. And we applied this algorithm for 18 healthy subjects of mean age of 70.22 years and male to female ratio of 1.25. As we can see from this graph, we get this expected CVR response, as we can see as other studies have reported earlier. And also, we could calculate the CVR time delay, and where we could see higher CVR amplitude in cortical regions and shorter time delay. And white matter and other regions has a little bit longer time delays. But these these shorter time delays were not always associated with the higher CVR amplitude, as we can see from the ordered subregions of the cortex, where the frontal regions has a higher CVR amplitude and putamen region has the lowest, but the CVR time delay did not follow the same pattern in the reverse order. So therefore, as a summary, we presented a novel robust CVR estimated pipeline to adaptively capture BH response or breath-hold response from data itself, irrespective of the subject compliance. Or CVR time delay is estimated relative to a respiratory signal, and the subject showed the agreement with previous similar studies. So advantages of our study includes the serious deviation may have been avoided, but we could capture the slight deviation of the compliance by the subject, and we did not need any recordings like end-tidal CO2 or any study design parameter to use in a regression. And the limitation included this expect the breath hold paradigm to be more symmetrical, like similar breath hold and baseline period. But on the other hand, if it is a little bit away from the symmetry, we can still use same approach, but by adding harmonics to the sinusoidal waveform. And I would like to acknowledge the funding sources, and thank you for listening.
All right, thank you. Very interesting talk. Is there any questions in the chat, Chris?
No questions yet.
Okay, so maybe I would have one question. So it's a very interesting method to get the CVR without end-tidal CO2. But have you compared within the same subjects, what would if the signal is if the values are comparable with end-tidal CO2, and if not, do you think that kind of validation would be required before using that method in a more widespread fashion?
Yes, that's an important point, but unfortunately, this data that I am handling right now doesn't have end-tidal CO2 recordings. It only has the respiratory bed signal recording, but that respiratory bed signal recording, respiratory pattern shows a nice agreement with the calculated CVR response, but it would be nice to have it compared with the end-tidal CO2 recordings in the future.
Okay, thanks. I have one more question in the chat. So may you comment on how differences in basal metabolic rate and CO2 production between participants impact your measurement and impacts between group comparisons?
We did a group comparisons and it will be reported later in a different presentation, but the other questions we did not study. That is an important point, but in this particular study we not consider. We were developing this method to calculate CVR without directly from whole data, so we were focusing on that.
Okay, thanks.